So I'm going to get back started again. Talking about now, I'm going to be talking about in the first part of the talk, I talked about how genetic variation influences gene expression levels and splicing and all of that. In the second part of the talk, I want to talk about things we actually care about, the phenotypes that and diseases that we're interested in. And so, how can we identify genetic variants that influence diseases and traits, and how do they exert their effects? And so, in this is genome-wide association studies. And the question is, can we learn biology from genome-wide association studies? And this is the figure I showed you before, where we have a genetic variant that influences some sort of cellular phenotype, which in turn influences a tissue phenotype, which in turn influences an organismal phenotype. And a genome-wide association study, for people who don't know, is at the level of the organismal phenotype. So you take cases and controls of some disease, or measure lipid levels in hundreds of thousands of people. And these association studies over the last maybe 5-10 years have identified thousands of genomic regions that contain genetic variants that influence hundreds of diseases and traits. And for the most part, we don't know how these genetic variants exert their effects. And the basic study design is to test for differences in allele frequency between cases and controls. And I'll sort of walk through this quickly. A genome-wide association study. Imagine this is the human genome here on the x-axis. And we have polymorphic sites, so positions that are variable between two people. So imagine here you can have either an A or some other base. Here you can have a G or some other base, call it a T, and so on and so forth. In a standard genome-wide association study, you would take this genetic variant and say, are the alleles of this variant associated with disease, yes or no? In this case, and on the y-axis here, that's getting cut off, is the negative log 10 p-value for association. So further up here, it's going to be more associated, or more strong statistical evidence for association. And at the bottom here is less strong statistical evidence for association. And so this genetic variant is not particularly strongly associated with the trait. This one is not either. This one's not either. This one is. And so this genetic variant, we would say, is a true statistical association for the reasons we mentioned earlier. It's possibly not the causal association, but this is an association where we know that this variant, or one nearby, influences disease risk. And the standard way to do this, and this is what this looks like, actually, if you do this on real data. This is, on the x-axis, is position along the genome from chromosome 1, 2, 3, out to 22. On the y-axis is this measure of association. And this is a genome-wide association study for retinal vascular caliber, so the width of blood vessels in the retina. And you can see there are a few positions here where it looks like there's a genetic variant somewhere here on chromosome 19. There's one here on chromosome 12, one on chromosome 6, 8, and 5, where there's a genetic variant that influences this trait. And the standard way to call this is you look at a p-value of 5 times 10 to the minus 8th, that's this dotted line here, to call something significant. And the reason that people have come to this conclusion of how this, what seems like a fairly stringent statistical threshold, is that a priori, you don't expect any of these to really be associated with the trait. And so you have a low prior on any given genetic variant, so you need strong evidence to convince you otherwise. And that's what these... So the reason this may not be the standard way of getting to this significant threshold, but the way I think about it is that what you're interested in is what is the probability that a genetic variant influences a trait. And so if you're a Bayesian, that requires what's the prior probability that the genetic variant influences the trait, and the evidence. So there's one component which is the evidence, and there's another component which is, how ridiculous is this? So there's the saying, extraordinary claims need extraordinary evidence. There's one way of thinking about this is, well, saying this genetic variant influences this trait is sort of an extraordinary claim, and we want to have really strong statistical evidence to make that claim. Sorry? You said you want to take into account the prior. Oh, yeah, so I meant. Yeah, so I meant prior in a statistical sense, but actually looking at the history, the reason people have come to this, this stringent statistical threshold is because it seems to work, in the sense that you take these things and now you go to a new population and you double check to make sure they show up, and they all do. Whereas in the early days, before people were doing this, you get something with a p-value of 10 to the minus 4, and you'd get really excited, and you go and you try and test in another population, it wouldn't show up anymore, and it would just, the signal would disappear. And so what people have done is they've come up with these really stringent statistical thresholds to avoid that, to say we want to be really sure that we're looking at something real before we invest any more money going after it.
But if you look at this from the Wolfroni correction point of view, say, given the total number of SNPs, do you get the same ballpark threshold, or is it really different?
Yeah, it's about the same. Yeah, that's like Bonferroni. Yeah, it's like Bonferroni for 1 million tests. It's the same ballpark. Yeah.
I've always been curious about this. Obviously, you use Bonferroni because you want to minimize the number of hits, right?
Mm-hmm.
If you were to use an FDR, the more traditional FDR approach, but to control the crossover rate, like let's say 5%.
Mm-hmm.
If you probably get 100, maybe 1,000 hits, what would be what's the rationale for using Bonferroni versus
Yeah, so I mean, the reason I avoided saying Bonferroni or correcting for multiple testing is because, so that's equivalent to saying I want to test the hypothesis that there are no genetic variants in the genome associated with this trait. And so then you test 1 million different things, and you set a p-value threshold of 0.05 times 10 to the minus 6, which is 10 to the minus 5 times 10 to the minus 8 to do that. Whereas really, I mean, you're not so this isn't really multiple hypothesis testing, right? This is testing 1 million hypotheses, which are all different in some sense. It's like, is that genetic variant associated with the trait is a different hypothesis than is that genetic variant associated with the trait? And so it's not, I don't think about it in terms of classical multiple hypothesis testing in that sense. But
to me, it's almost like you're kind of using a Bayesian type of framework without actually using a Bayesian framework. So you're saying, well, there shouldn't be too many, too many hits.
Yeah.
You're basically using the theory, the assumption is that there shouldn't be too many hits. But
Yeah, exactly. Mm-hmm.
This might be a very stupid thought, but given that usually an association signal has multiple SNPs when linked to each other that are both have a big p-value, right? And we treat them as independent when they're really not. So would that make our threshold more stringent than it practically needs to be?
Right. So, in practice, what people, so I think there's no actual, I would not call this 5 times 10 to the minus 8th like it's sort of like an empirical rule of thumb that has seemed to work in the past. And you can come up with sort of justifications for how to get there, but the fact of the matter is that it's just that you take these and you try to reproduce them, and they reproduce. And that's why people have come to that conclusion. And it's sort of a good rule of thumb if you just did a random association study and you wanted to just see if there's anything there. It's a rule of thumb based on sort of the history of the field that if you want to be really sure that there's something there, this is what you do. But it's not it's not set in stone. And the statistical justification, as I was saying, is sort of, should it be 1 times 10 to the minus 8? Should it be 5 times 10 to the minus 7? Yeah. Doesn't really, there's no real good statistical justification for any of those. Just something really stringent. And so, in any case, that's what this looks like. This is a genome-wide association study. In this particular case, they found 5 loci that appear to contain variants that influence this trait. And so the standard way to do this is to treat all SNPs, these are single nucleotide polymorphisms, treat them sort of identically. So the only thing that matters is that this is a polymorphic site, and that's a polymorphic site, and that's a polymorphic site, and that's a polymorphic site. And that's, from the point of view of your statistical software or whatever, that's what matters. Polymorphic site or not polymorphic site. But we know the genome is actually more interesting than that. We know there are things like exons that code for proteins. And if we maybe put these in blue, this SNP is not just a polymorphic site, it's a polymorphic site that falls in an exon, right? Or maybe there's transcription factor binding sites, and we know where transcription factor binding sites are. And this SNP is not just a polymorphic site, it's a polymorphic site that falls in a transcription factor binding site. And maybe that changes the way we think about it. And actually, in this case, in this toy example, that's where the true statistical association is. And so, can we relax this assumption of every site in the genome is sort of equally plausible? And if we relax that assumption, what can we gain? And that's sort of been a lot of my work over the last year and a half, is trying to play with this assumption. And so why would we relax this assumption? Just to sort of go through the evidence. We know that things associated with the traits are actually enriched among non-synonymous SNPs, which makes sense. If a genetic variant changes protein sequence, it's more likely to affect a phenotype, and we know that. We've known that for about 5 years. We know that SNPs associated with traits are enriched among EQTLs. Dan Nicolai, I think, was one of the first to show this in 2010. Thule showed it last year. And SNPs associated with traits are enriched in cell type specific regulatory regions. So if you do a genome-wide association study for, I think the example in Trinka et al. Was a lipid type trait, you find that they're enriched in regulatory regions that are found specifically in, say, liver. And so we know that we should be able to relax this assumption to some extent. And why would we come up with a new statistical model for this? You want principal ways to deal with LD. So LD is this correlation of nearby SNPs in the genome and multiple annotations. So you might want to say, well, are SNPs, are non-synonymous SNPs and enhancers enriched for genome-wide association studies, whereas the standard ways to do this now would take each of them individually. And some questions just can't be answered with existing approaches. For example, imagine we had 2 SNPs with a p-value of 1 times 10 to the minus 7th, which is considered non-significant based on the argument from before. And so one of these is non-synonymous, and one of them is far from all known genes. And then I asked, what's the probability that each of them is actually a true association? A priori, you might say that the non-synonymous one is more likely to be a real association, even though it has the exact same p-value. But that's an intuitive way of saying we're doing Bayesian modeling. If we have a prior and we're adjusting it in light of some evidence. And so the goal of the statistical model I'm going to talk about is, so the fundamental question is, what are we actually interested in when we perform a genome-wide association study? One, it's what is the probability that a given part of the genome contains a SNP associated with a trait. We're not really interested in the p-value. We're interested in the probability that there's a SNP there that influences the trait. And if a given genomic region contains an association, what's the probability that any of them is the actual causal site?
So is it fair to say if you imagine like a dream of having a full mechanistic model of how the cell works, in your model would give 0 probability to all the non-causal SNPs and then probability 1 to the causal one, and you're kind of building up to that by trying to capture some trends?
Yeah, exactly. Like, it's if you saw a SNP, could you say this genetic variant influences the transcription of this gene and influences height? Like, could you say that? Or imagine I gave you a random genetic variant. What can you tell me about it? Without knowing anything else about it except where it is, and maybe some functional genomic data. That's sort of like the high-level goal. And so the basic building block, this is the statsy part of the talk, is just a basic regression model. We have the phenotype of some individual, and we're going to model it as just a linear regression that depends on where did that go? There's something there that said genotype of individual I, and so it depends on the genotype. So the phenotype depends on the genotype, and we're going to use so the standard way, or a standard way to look at this is to calculate a p-value for association. I'm not going to calculate a p-value. I'm going to calculate a Bayes factor, which is slightly different, which is the probability of the phenotype data given the genotype data if the SNP is associated, divided by the probability of the phenotype data given the genotype data if it's not associated. So it's just a ratio of these two probabilities, and I'm integrating out beta and alpha. So I don't really care about beta and alpha. I just want to know what's the probability of the data if this is not 0 versus the probability of the data if this is 0. And so there's actually a trick in the literature to compute this. John Wakefield in 2008 pointed out that if we say this is the observed summary statistic, so if we take this to infinity, we have 100,000 people, and we've observed their phenotypes and their genotypes. We don't actually care what the phenotypes and genotypes are. All we care about is this. And so we can just pretend this is normally distributed, which it is, and the limit is the amount of data goes to infinity around the true regression coefficient and some sampling variance. And then the true regression coefficient we model as normally distributed around 0. So most of the mass is around 0, and there's a bit of a tail. And so the reason to do this computational trick is that if this is 1 million people and that's 1 million people, and you want to do this 1 million times, now you're counting to 1 million a lot of times, and it gets very computationally intensive. Also, in the field of human genetics, unlike in other fields, people don't share their data as well, but people are often not willing to give you genotypes and phenotypes for 100 million people, 100,000 people, or 1 million people, but they're often willing to give you the estimates of beta. So just if I took a linear regression at all 1 million sites in the genome and estimated this parameter, people are more happy to share that rather than sharing the raw data with you. But it turns out you don't really care about the raw data if you do this quick little trick and you model the observed regression coefficient just as normally distributed around the quote-unquote true regression coefficient.
So this, just make sure that I understand this. So you want to know what is the error in the slope of your regression, right? And then just to connect it to something we talked about, B is like the estimate of the standard deviation, like 1 over n minus 1, and then W would be more like our sigma, right?
Yeah. So the sampling variant in this particular case, the sampling variance in theory is 1 over 2n p 1 minus p, where n is the number of individuals and p is the allele frequency. So it's the binomial sampling variance.
What is the trick here now exactly? I'm not sure I got that.
So the trick is to forget about the genotypes and the phenotypes and pretend, actually, so imagine someone just gave you p-values. Someone gave you 1 million p-values and you wanted to write down a model imagining you had the actual genotypes and phenotypes that generated those p-values. And the trick is to note that as the amount of data goes to infinity, this goes to normal. And so now you can just, instead of computing this, you just compute this under beta equals 0 and beta equals not 0. And you can take those, so I want to estimate this, which is the probability of the data if there's an association and the probability of the data if there's not an association, and the ratio of those. But I don't have the data, is the problem. So it's difficult to estimate the probability of the data if you don't have the data. So that's this trick.
What do you have? So just because I want to make sure that we're on the same page. So for instance, this is a GWAS kind of thing, right? There's no expression. So G would, for instance, be 0 if you have the major allele, and 1 when you have the minor allele, right?
Yep.
And then Y would be the
Height. Yep.
Or some individual. So with major allele we have height alpha, and with the other allele we have alpha plus beta.
Yep.
Right? But then, if you don't have the data, what do you have?
Yeah, I'm going to say I have the p-values.
Okay.
So someone has done this through some complicated procedure and just gives me 1 million p-values.
Oh,
for each genetic variant.
For each genetic variant.
Yeah.
But is the idea that the beta is the same for those different variants?
Right. No, so the p-value is related to the beta in some way that I can sort of figure out. I can sort of back calculate.
Right. See, because I try to understand that it seems like basically the p-value is something like the area, the cumulative distribution, right?
Yeah.
The beta. And I could imagine if you have a bunch of p-values, you can kind of go back to the z-score or something, right? So I can imagine that from that you could estimate like the width of. But if you have p-value per trait, you have only one number from that distribution, then I'm having a hard time
Yeah.
understanding how this works.
So I have this computed for each genetic variant. So I have genetic variant 1, I have a p-value. Genetic variant 2, I have a p-value. Actually, I have this. Imagine I just had all of these p-values.
So one trait and then
Yep. And then the whole genome worth of p-values. Each one of these comes from a separate SNP. So this is one. So in this case, I don't know how many people. Let's say 10,000 people on a genotyping array, and you measured retinal vascular caliber or whatever. Yeah. So if you don't want to think about the computational trick, just imagine I can calculate this. Like, imagine I had all of the genotypes and the phenotypes. In some cases, you might actually get genetic data from 100,000 people and phenotypes from 100,000 people instead of this, summary of what happens when you do that. You just have to be able to compute this somehow. And the particular way I'm going to compute it is to use this, where the observed regression coefficient is normally distributed around the truth. And under these assumptions, the Bayes factor is this, which just depends on the z-score, like you said. So it's only a function of the z-score and the variances. And that's all. So if you don't want to worry about the trick, just imagine you compute that somehow, and now you need to plug it into some sort of model. And so the model I'm going to do is I'm going to split all the genome up into blocks, so K large regions. So this is to get around this issue that was mentioned before, that nearby SNPs are all correlated, and we're treating them as independent. So I don't want to do that. So I'm going to take 2.5 megabases and take each 2.5 megabase chunk and treat each of those as independent, which is they're still not totally independent, but they're definitely more independent than 2 nearby SNPs. And what's the probability that each region contains a single association? Single causal association, so they can have multiple statistical associations to get at that. And the probability of these betas is just going to be the probability that the block has an association, and the probability of the data under that block, under that model, and the probability of the data under the alternative model times the prior probability that that block contains an association. So split the genome into blocks and say, does this block contain an association, yes or no? Does this block contain an association, yes or no? Does this block contain an association, yes or no? Out to the end of the genome. This is like a model that Jean-Baptiste Verrières and colleagues developed for the analysis of EQTLs, actually. And so now we can play around with this. So what's the probability that any given SNP is the causal one in the block? It's the prior probability times some probability. But this is the key, that these prior probabilities can depend on external information, and you can estimate the values of these parameters. So here's the prior probability that any block has an association. It's going to be something plus the effect of an annotation and whether that block has that annotation. So for example, is this region gene dense? So I've gone along the genome and I've said, does this block have an association, yes or no? Does this block have an association, yes or no? Does this block have an association, yes or no? And maybe that can depend on whether this block actually has 100 genes in it, this one has 0 genes in it, this one has 1,000 genes in it, and maybe that matters to me.
So basically what you try to do here is to have some kind of uniform, homogeneous way to combine the p-value from the SNP association with the context feature kind of weighting, right? So it's almost like you have a protein complex forming on DNA, and it's all these energy.
Sure.
Because
Yeah.
the wave factor is like an energy, right? You're trying to compute the probability that you form this complex. So you want some protein-protein interactions and some protein-DNA interactions, right? In a way, that's how you want to integrate these things.
Yeah, exactly. I want to know, does this region of the genome contain an association? And I think it matters to my belief whether or not that region of the genome is full of genes or not, a priori. But then we can let the data tell us that. We can learn this effect size, and maybe actually it doesn't matter. But I want to test whether or not that matters. And so that's what these annotation parameters, which are actually learned from the data, can do. And so that's the effect of the region. And now, what's the prior probability that any SNP is a causal SNP? It depends on, it has this functional form again. But again, it's going to depend on things. So now I've gone into this region. I say, this region of the genome has a SNP that influences the trait. Well, what's the probability that it's that one, that individual SNP, or this individual SNP, or that individual SNP? And I can say, well, this is a non-synonymous SNP. Maybe I should upweight that one a little bit. Like this, like the example we gave before, we have two SNPs with the same p-value. The non-synonymous one, maybe we think is more likely to be the real causal SNP. But again, we don't want to impose that outside. We want to learn that from the data. And so we want to we're going to learn this parameter, and the data is going to tell us if we should care about non-synonymous SNPs or not. And then we can put this all together and just write down the likelihood of the entire model. And it depends on the prior probability of an association, and then the sum over all the prior probabilities of the SNPs and their Bayes factors. And yeah, so it's saying, imagine I took a random SNP and asked you, do you think this is the causal SNP? Let's say there are 5,000 SNPs. Maybe you would say 1 out of 5,000. Your probability is that it could be any of those 5,000 equally, if you don't know anything else. But let's say you know that the SNP in the middle is non-synonymous. So maybe you don't want to call all of them 1 over 5,000. You want to go drop all of them a little bit and maybe give that non-synonymous one 3 out of 5,000 or something, a priori, without looking at the actual association data. So if I randomly took a SNP and told you it was non-synonymous, and then I had you guess whether or not it had an actual, whether it actually influenced height, would that depend on whether I told you it was non-synonymous or not? That's sort of the relative prior probabilities. And I sort of mentioned that we can learn all of this from the data, which seems sort of circular, right? I told you this is a prior probability without looking at the data, but I'm going to learn it from the data, right? That seems a bit circular. And so it's going to be slightly overfit. And so I'm going to actually use a penalized likelihood in cross-validation. So this is the likelihood, then I'm going to penalize it with these annotation effect sizes and subtract that off. And this is a penalty used in, say, ridge regression and some of these machine learning techniques. And so that was just sort of flying through the model. It seems all fine in theory. Let's see what happens in practice. And so what I'm going to tell you about are 18 genome-wide association studies with publicly available summary statistics. And that's why I spent all that time talking about this computational trick, is that I could just download the summary statistics off the internet without writing 10-page applications telling people exactly what I'm going to do with their data. And then I imputed this to all of the 1,000 genomes data. And this is a bunch of association studies of lipid traits, height, BMI, bone density, Crohn's disease, red blood cell traits, and platelet traits. And then I'm going to look at 450 genomic annotations. So 400 DNase hypersensitive assays and hundreds of tissues. And then the ENCODE project has segmented the genome into sort of repressed chromatin. This is sort of a computational analysis of chromatin marks. And they've partitioned the genome into repressed chromatin, enhancers, transcription start sites, promoter flanking in 6 cell lines. And then I have gene model, so where are exons, where are introns, what are non-synonymous SNPs, what are synonymous SNPs. And so then I have 450 functional genomics data sets, 18 genome-wide association study data sets, and then you can ask, for each of these genome-wide association studies, what actually matters? Should I be looking at DNase hypersensitivity in the brain if I'm interested in lipid levels? Should I be interested in DNase hypersensitive data in the liver, or should I be interested actually in just in exons? And so here's an example for HDL. What I'm showing you here is I've taken all 450 genomic annotations and I've ordered them, so this should go down to 450 at the bottom here. And I'm just showing you the top, like the most informative associations for high density lipoprotein levels. And what you can see So over on this side are things that are enriched, and these things are depleted. So where do SNPs that influence HDL levels fall? And they tend to avoid chromatin that's marked as repressed in HepG2 cells. This is a liver carcinoma cell line. They're enriched among transcription start sites in HepG2 cells. They're depleted away from K562 cells, repressed chromatin K562 cells, et cetera, et cetera, et cetera, et cetera. And so just by plugging in the genome-wide association study, we've learned something, at least, about HDL levels, which is that the liver matters, which maybe we knew that already.
Can you quantify, if you think of the total, the analogy of like adding up the energies, right, and then it gets a probability of that this is really a functional SNP, right? What part of this accumulation of evidence, right, in this kind of energy-like scale, is due to these context features as opposed to just the association, right?
Yeah.
The GWAS association. Because in a way, once your model gets fancier and fancier, at some point it's going to be more than 50%, right? And then, and again, ultimately, you would like it, just the model would be able to predict this.
Yeah.
How transcription factor binding is, works and chromatin context and all that. Then you don't need that.
Yeah. If you knew that this SNP influenced this transcription factor binding site, which is near this gene, and this gene is really important in lipid metabolism, then you might be able to say, without knowing anything about the actual association, that this SNP must influence lipid metabolism.
So how far away are you from this?
I think we must be really, really far. But I'll show you a plot that maybe has something to do with that. Yeah.
What with the color codes here? What's red?
Oh, yeah, sorry. So I'm going to show you on the next slide, I think. So this is taking each thing individually, and a lot of them are really correlated. And so what if you just tried to build the best? What if you add 2? What if you add 3? What if you add 4 of these all in? And do they keep getting significant? The red ones are the ones that stay after you've tried to build a joint model with multiple things. And in pink are things that are statistically indistinguishable from the things in red. So I could put in non-synonymous SNPs or coding exons, and statistically they're indistinguishable. And black are things that fall out of the model. Yeah.
You're not using gene information, right? So what
Right.
Harlan said is
No.
you would be able to say where it is, but you're not using the information in the gene. I mean,
That's right.
type of it.
Yeah,
So you
I'm
would be able to know what type of SNP it would be,
Yep.
but not which one.
Right. So yeah, a non-synonymous SNP in a gene that's only expressed in the brain is considered a non-synonymous SNP here, just like a non-synonymous SNP that's expressed only in genes expressed in the liver. Yeah. So one clear area of improvement would be able to sort of partition that out into things that we think do different functions. Mm-hmm.
So you mostly examine intestinal / the hepatic tissue, which is relevant to lipid metabolism here. If you did the same types of analysis on cells that are not relevant to the biology of lipids, do you see what they claim, that a lot of the GWAS falls in tissue-specific enhancers around the liver? Would that, if you do the same type of analysis in a cell type that is not really relevant to lipid biology?
Yeah. So a lot of these things are really highly correlated. So here, fetal muscle, actually, so this is the enrichment of DNase regions in muscle. And that's it's pretty highly enriched, actually, even though muscle has nothing to do with, as far as I know, with lipid metabolism. But this is actually correlated with probably one of these here, transcription start sites in HepG2 cells or something. So there's a lot of, tissues aren't independent, right? So the muscle probably shares some gene regulatory pathways with the liver and so on and so forth. And so these things aren't totally independent. And so I'm not sure I'm answering your question.
I guess I didn't notice the muscle. There's the muscle can serve as an example of a
Yeah.
non-relevant.
Yeah. And so I'll show you an example later where I think I have it, where it's SNPs that influence height are enriched in chromatin that is active in the lung or something like that. And so how would you interpret that? I don't have a great interpretation of that. One potential interpretation is just we're missing the right tissue. Like I don't have the right tissue for height in the data. Maybe it's the growth plate at some particular stage of development, which is really important, and that's just not in the data, so the statistical model can't do anything to find it. Yeah.
Or oxygen?
Yeah.
K562, where are they from?
They're erythroblasts. They're red blood cell precursors.
Mm-hmm.
And so, like I said, okay, so now we can generate a combined model. This was taking each thing independently. So this is the algorithm to generate the combined model, find everything that's significant if you test it one at a time, and add it. And so you force in a bunch of other things. And then do this forward selection, which statisticians tell you is a terrible idea, but I'm going to do it anyways. Throw in the annotation, try all the other ones, and then try all the other significant ones, add the best one, where best is defined as the one that maximizes the likelihood, and then iterate. So I've thrown in HepG2 cells. Now I'm going to test HepG2 cells plus everything else, and then so add something else in, and then try all combinations of 3, and then all combinations of 4, and so on. And then I'm going to use this penalized likelihood and cross-validation to try and shrink everything back down to make sure I'm not overfitting the data. And then I'm not going to talk about that really.
How does what you're doing compare to, so you're doing all this in the Bayesian framework. How would this compare algorithmically? How could you use penalized regression to do something similar? I mean, you come at it from, you build it up with a Bayesian model, but when you describe it here, it's beginning to sound like a regression probably,
Yeah.
Where you're basically trying to predict p-values, essentially based on features that are nearby or based on annotation.
Yeah. So it's sort of a regression model, right? So I'm building up these effects of these annotations, and then adding in a new annotation, adding in a new annotation. Yeah. But it's the output is not the p-value per se. It's it's like a real likelihood sort of. And so I guess there's probably a way to reconstruct it that it's not in a Bayesian framework, but I haven't done that. I think it's more intuitive to think about what's the probability that SNP influences a trait rather than what's the probability of the data if the SNP does not influence the trait, right? I guess is.
So you're trying to compute the posterior distribution of the effect size, right?
I've actually regressed out beta completely.
Oh, really?
And all I care about is the probability that the SNP influences the trait.
Is there a way to integrate the QTLs in this?
Yeah, that's a great question. Probably. That's something I'm thinking about how to figure out how to do. Yeah. I think that would be really useful. And so this is the combined model I mentioned before. If you build up a model for localization of GWAS hits in HDL levels, and you combine everything, this is what ends up being important. So gene-rich regions, that's a 2 to the 4th-ish, or so, 2 to the 2, so 4 times enriched in gene-rich regions. They're 8 times enriched in coding exons and transcription start sites in HepG2 cells. They're depleted, 4-fold depleted from repressed chromatin in K562 cells and HepG2 cells. And each of these seems to have approximately an independent effect. And so with that, we've sort of learned a little bit about the biology of the trait. Imagine we didn't know anything about lipid levels. We learned that HepG2 cells are a good proxy for something that's important about the trait. So liver cells are somewhat important for HDL levels. And so what if we do this for all 18 traits? So if we start going through all of our traits, here's mean cell hemoglobin, and here's the same sort of model now for hemoglobin levels, and we find that enhancers in K562 cells are about 4 to 8-fold enriched among the GWAS hits. So the GWAS hits are 4 to 8-fold enriched among K562 enhancers and depleted away from repressed chromatin in K562 cells. And that was your question before, was what is a K562 cell? It's a precursor to red blood cells. And this is hemoglobin levels is our phenotype here. So again, we've learned something. If we didn't know anything about hemoglobin, we've learned that K562 cells are a potentially relevant cell type if we were interested in the biology.
So you said a threshold in terms of the GWAS hits. That was the initial threshold that you.
Yeah, so I'm not really setting a threshold.
You know.
There's a probability that a segment of the genome influences the trait, and for most of the genome, that's 0. It ends up getting set to 0 in the algorithm. I'll show you.
I was going to say, I mean, that could also allow you potentially to test different thresholds and see whether, allowing more hits
Yeah.
would actually potentially give you a better fit.
Yeah. I'll show you an example of that. And so another thing I thought was kind of interesting was this repeated observation of repressed chromatin. So here's LDL levels, for example. SNPs that influence LDL levels are preferentially non-synonymous in near transcription start sites, and they're also far away from repressed chromatin in HepG2 cells. And here's height, as I mentioned before. They're enriched among open chromatin in the fetal muscle and the fetal lung, and they're depleted away from repressed chromatin in HeLa cells. And so what this is sort of saying is that for a lot of these traits, we don't know what's important, but we do know what's not important, and that's repressed chromatin. Yeah.
But isn't the very nature of repressed chromatin that you would expect that SNPs associated with traits and that have an effect, a phenotypic effect, would not be associated with repressed chromatin?
Yeah, I guess if you stop and think about it, then yeah. But the way these things are defined is just in terms of coordinated chromatin marks. And the way the field has sort of thought about this is where are things enriched, not where things not unenriched. Because it's a sort of a weird thing to think about. But yeah.
It's a nice verification, and it actually gives strength to the enrichment values to be able to say
Yeah.
that what you would expect is not enriched.
Yeah, sure. The model could have inferred this to be up there, right? And then I would have started to worry a bit. Maybe if repressed chromatin was 100-fold enriched for SNPs that influence traits, yeah. But another way to sort of think about it is that a repression or a depletion away from repressed chromatin is an enrichment among everything that's not repressed, in a way. So maybe we don't know everything that's going on in non-repressed chromatin, but we know something important is going on there that's relevant for height and LDL levels, at least. And so in some of these, like I said, hypersensitive regions, so these particular sort of enhancer marks in relevant cell types are often enriched for associations. These are so this is for a GWAS of platelet volume. This is a GWAS of platelet count. And what you can see is there's a clear enrichment in both and CD34 positive cells, which are the cell type that's the megakaryocytes, which give rise to platelets later in development. And so you can, again, you're sort of learning hopefully interesting things about the biology of the trait, even though in my case I knew nothing about platelets, really. And sometimes these things don't totally make sense. So here's platelet volume again, zooming in. We have CD34 positive cells, enhancers in HeLa cells, maybe that makes sense, transcribed regions and lymphoblastoid cells. But here's the spleen. The model seems quite confident that genetic variants that influence platelet count are enriched in the fetal spleen. And I don't have a great explanation of that.
What's the number in
Yeah, so the number in parentheses is the number of other. Remember, I had these things in red and the things in pink, and the things in pink were things that were statistically indistinguishable. Like I had exons and non-synonymous SNPs, and the model could call something an exon or an exonic SNP, or could call it a non-synonymous SNP, and it didn't really matter which. So these are correlated. It's the number of other things that are highly correlated with this. So there are 35 other things that are really highly correlated with enhancers in HeLa cells. Yeah, that predict equally as well. And so one is there's nothing that predicts as well as the fetal DNA hypersensitive sites in the fetal spleen. Yeah.
It's interesting, the fetal spleen thing. There are several human conditions that where the spleen can become enlarged and can lead to splenic sequestration.
Mm-hmm.
And so the platelets can get stuck in the spleen, and you can end up with patients the platelets dropping to the point that
Yeah. The spleen is involved in platelet biology, but then, so you start to wonder, these are primary cells dissected from an actual fetal spleen. Maybe what is this actually measuring? Is it measuring spleen, or is it measuring the platelets that are already in the spleen, right? So the model's only as good as the data that goes into it, right? Yeah.
So were these all the sort of annotations that were included with this like GWAS test for platelet volume? Or
Right. So this is, if I took all 450 that I started with, and then used this algorithm to collapse them to the ones that matter for platelet volume, and that's what's left out of the 450 that I started with.
I see. So the things that aren't listed here are the ones that you have like not.
Yeah, they just dropped out. Yeah. Yeah.
It looks like that 3' UTR in the list actually.
Oh yeah.
Yeah.
All right. Yeah.
Just where you say that you're saying where it maybe enhances because of the 35 of the variables that could give an equal blah 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 in the model, how did you choose to represent that rather than one of the others? Or did you just choose randomly?
So I chose it as, so it's the best of those 35, and that it improves the likelihood of the model more than the other 34, but not significantly so.
So there might be actually 34 ones that might be better in this list than the fetal spleen DNA.
So no, there's.
No, but I mean all these 34 other enhancers.
Oh yeah. But they're not correlated with fetal spleen. They're correlated with enhancers and HeLa cells.
But
Yeah.
they might have been better in the model than one of these other variables
Mm-hmm.
on their own.
Yeah, that's right. That's right.
Just out of interest, if you refer to repressed chromatin, do you put heterochromatin and polycom repressed?
Oh, from the segmentation algorithms?
Yeah.
Yeah. It's everything is thrown into one repressed state.
Would you think it would be maybe different if you would separate polycom repressed chromatin? Because I guess the idea being that it's maybe more dynamic in terms of
I see.
yeah.
I haven't
Out of
looked. I
interest.
should look. Yeah,
Because I
that's a
guess
good point.
heterochromatin wouldn't change much, but maybe polycom repressed regulatory elements would maybe
I see.
potentially.
Yeah, I'll try that. Thanks.
Sorry, that's a question. One of the things that you did initially was to pick 2.5 megabases as a region
Mm-hmm.
to look at. And I know that's something that's widely done in SNPs, that you look within a locus or within a region of chromosome. Did you play around with that any bit? Did you get any differences? I was just wondering, particularly when you're looking at these gene-rich and gene-poor regions, particularly chopping it up into segments like that, you may be in a boundary or something like that. So I was just
Yeah.
wondering, did you look at overlapping regions, or did you try different sizes?
I tried different sizes.
It's an interesting thing for the field because I know it's something that a lot of people do.
Yeah, so I've tried different sizes in a handful of these, and they didn't seem to change much, so I didn't do an extensive analysis. One thing I'm interested in doing is there should be, I think in principle, I haven't shown, in principle there should be a principled way to split up the genome based on the patterns of correlate. So the reason I'm splitting up the genome into blocks is to get rid of this correlation structure. But we know the correlation structure of the genome to some extent. So in principle, you should be able to split it up into blocks in some optimal way to do this, instead of just taking 2.5 megabase chunks, which is sort of just a hack. But yeah. Mm-hmm.
Do you have any features like these that keep showing up over and over across phenotypes?
Yeah, non-synonymous SNPs, and various repressed things. But yeah. And so, yeah, go ahead.
So one question, I'm kind of interested, you have a nice tool now to see whether, for instance, blood cells are at all useful for understanding like brain traits,
Mm-hmm.
right? And it doesn't matter whether it's above or below in the enrichment.
Mm-hmm.
So what is the general picture there?
So the only brain trait I've looked at is schizophrenia, and none of the blood things really show up. There's some DNase in the fetal brain, which is the only thing that shows up. But, telling you that the brain is important for schizophrenia is not extremely useful. But, yeah. And so this is what the question just was. So are there any things that we can sort of one thing I was kind of interested in is whether we can test for, take one particular annotation and test it across all 18 traits. And so non-synonymous SNPs are definitely associated, enriched for associations for all 18 traits to some extent. And so these here and that one are not statistically significant, but they're all in the same direction, including the ones that are statistically significant. And this is the level of enrichment, which ranges from around, oh, this is natural log here, so 3-fold to 10-fold. And actually, in this model, you can turn this around and ask what fraction of the hits are actually non-synonymous. And since non-synonymous SNPs are so rare, even a 10-fold enrichment doesn't correspond to a ton of SNPs. And this ranges from about 2% up to 20% or so. So as people have noted before, the vast majority of what's going on in terms of genetic variants that influence phenotypes is happening at the level of gene regulation. It's not happening at the level of changes in protein structure. We sort of see a maximum of 20% of the hits here are driven by changes in protein structure, leaving, of course, 80% to be due to other things.
So can you go back to GWAS hits and like re-rank them using this? I guess we have that, you have a set of rules within the kit to tell you whether a hit is more likely to be functional.
Yeah, exactly. And so here's, so let's go back to this combined model for HDL cholesterol, which I showed you before. Well, what if we wanted to re-rank the genome-wide association study? We can write down, instead of this Bayes factor for the SNP, we can write down the Bayes factor for the region, which is just multiply together the prior and the Bayes factor for the SNP. That's the Bayes factor for the region. The posterior, compute in the same sort of way. It's just the posterior prior odds on the region times the Bayes factors gives you the posterior odds for the region. And what does that look like for HDL levels? So here's, each point here is a region of the genome. And I'm plotting the posterior probability of association, so the probability that there's an association in the region, under the model without any enrichments, and under the model with enrichments. So under that model I showed you before. And what you can see is there are quite a few points up here above the line. In blue are regions where the minimum p-value is less than 5 times 10 to the minus 8. So these are sort of standard genome-wide significant regions. In red, the regions where the posterior probability of association is over 90%, but the minimum p-value does not quite reach standard genome-wide significance. And so these are regions where the functional genomic information is adding something a lot. And it's saying, previously I didn't think this was a real association. Now I think it is a real association. And it's because of these sorts of functional genomics enrichments. And so let's look at one of these examples. So this is a genome-wide association study of HDL levels. And along the x-axis here is position along chromosome 1. And the y-axis up at the top here is the negative log 10 p-value. And you can see a peak of associations. And I've highlighted one SNP here, RS something something something. It had a p-value of 1.5 times 10 to the minus 6. And so this is obviously well short of your standard 5 times 10 to the minus 8 significance threshold. Actually, there's nothing here less than 10 to the minus 7 or so.
So the prior 1%, should you think of that as a much lower prior that got boosted by having a good genomic context?
Yes.
The 1% threshold?
This is, so yeah, so here's the prior for each of these SNPs that the model learns, right? And so this SNP that I highlighted before, for reasons that will become obvious, falls in this peak of prior. And so actually it's a prior greater than 1%, conditional on there being an association in the region. So it's actually, 1% divided by 100 or something. So it's 0.001%, which is still really high.
Because in the very beginning you were mentioning you assumed a very low prior, right?
Yeah.
Across the genome. So it's like 10 to the minus 4 or something?
Mm-hmm. Yeah, exactly. And so, yeah, the important thing here is that more like the shape of this prior, and that it's peaked on particular things. And the reason it's peaked on here is because it's not, so I guess you can't really see, but it's not in repressed chromatin, and it falls in a coding exon of a gene that's near or in a transcription start site of the of a gene expression HepG2 cells. So this is, according to the model, this is a particularly good candidate for a SNP to influence HDL levels, and it has a decent p-value. 1.5 times 10 to the minus 6 is not something you'd write up, but you'd think you'd take a second look maybe, and the model's telling you to take a second look. And actually, in this case, as I was writing this up, the paper came out, and the International Lipid Genetics Consortium, and now has a p-value of 9 times 10 to the minus 16th. So this is whoppingly significant now because they've doubled their sample size. And so but I would argue that if you had used the functional genomic information, you already knew that this was a really strong association. It's a non-synonymous SNP and a gene called NR0B2, which is thought to be involved in lipid metabolism, and now there's strong evidence that it is involved in lipid metabolism. And so, just to sort of conclude, this is a new statistical model for the joint analysis of genome-wide association studies and functional genomic data. And you can model multiple annotations, and it accounts for this correlation structure, at least to some extent. The repressive chromatin marks in specific cell types are depleted for GWAS hits. DNA-sensitive regions and enhancers and non-synonymous SNPs are enriched for GWAS hits. And so, just to tie this back to the first part, I talked about how does genetic variation influence traits. I told you about combining GWAS with functional genomics information can identify the relevant cell types. That's the association of the organismal phenotype and the cellular phenotype. I told you about EQTL studies that's down here, looking at the cellular phenotypes. So obviously, as was mentioned before, the next steps actually explicitly combining these different levels of information to try to put together this causal pathway from cellular phenotype to tissue phenotype to organismal phenotype. And with that, I'll stop and thank the EQTL work was during my PhD with Jonathan Pritchard and Yoav Gilad, and the GWAS work was recent, and thanks to all the consortia and ENCODE and Roadmap Epigenomics for making their data publicly available. Thanks.
How many of these fall out of the picture and how many fall? How does it change? Like, where are the numbers like?
Yeah. Oh, I think I have this. So what do you gain in terms of just numbers of loci? And so this is the lipid data, and the reason I'm using the lipid data is because there was a previous genome-wide association study that was big that I did this analysis on. Now there's a new genome-wide association study which is even bigger, and so you can sort of like compare. What if you only knew about the early one? Would you predict what happened in the later one? And so what did I do? I ranked all of these regions according to the p-value and according to the posterior probability of association, and now look in the new data, and you don't gain a whole lot. If you look just at the p-value, you would gain, so here's the number of false positives, where false positives is just things not found in the big study, and the true positives are things found in the big study, so they're not really false positives and true positives. There could be some of each, but if you rank things just based on p-values, you get 44 loci for HDL, and the numbers here don't quite add up because the large study didn't use as dense of a genotyping chip, so some regions aren't included here. If you use this posterior probability of association, you get 45 loci. If you use either, you get 49 loci. So you get, if you were to use both of these approaches, you would get an extra 5 loci.
Are they the same loci?
Yeah. Yeah, so the 45 is a superset of the 44, and the 49 is the sum of the both. And so, so I was negative about this. I said you only get about 5 extra loci. Someone else who's more optimistic said you get 5 extra loci. That's 10% more hits. That's, another 50,000 individuals, which costs another $1 million. So it depends on how you think about it.
But do you think that using functional data to maybe fine map those loci would be an interesting application?
Absolutely.
If you do that experiment
Yeah. So
Like how much closer do you get?
yeah. So one way, the way I think about sort of framing this question is imagine you had to functionally test, X number of loci. Um, and now you do this, sort of re-ranking and all of that. How many SNPs do you have to test now? And it ends up being something like you gain 10%, 15%. It's not huge on average. So there's some individual loci where the model is sure that it's one SNP, but that's actually relatively rare. It's usually quite diffuse. It's less diffuse than just staring at the p-values, but it's still quite diffuse. And so one possibility is we don't have the right functional genomic information. The other possibility is just that, it's going to take a lot of work. But I mean, yeah.
So if you try to, let's say, there's these things that you can explain like 8% of the variance in height, in terms of the genome or something, right? Does your weighting by context features help you explaining more of this variation? Or I could think, on the one hand, you think you find more SNPs, but you still, if you try to build a predictive model, you're going to get the coefficients from the actual trait data, right? So from that point of view, it wouldn't really help, it seems.
Yeah.
Is there a way in which it could help? Unless you have individual specific functional genomics DNA stickers.
Yeah,
But
great
if
point.
you don't, if you only have like a reference thing, then
Yeah, I don't think the gain would be huge, but I haven't looked at that specifically. That's a good point.
So during the re-ranking process, I guess only so additional SNPs like became more sort of supported by you didn't see the opposite like SNPs that were kind of harvesting based on just p-value and then kind of went down because they were not in the right context.
So no regions went down. Sometimes the SNPs get sort of like if the top SNP was in repressed chromatin, it would probably get bumped down a little bit, but that in turn would bump up a SNP that's in LD with or something. Yeah.
one more kind of technical thing is if you get an association somewhere, you know that somewhere near within that kind of statistical neighborhood of that SNP, there has to be a causal SNP, right? If you sequence everything, you know it's there, right? So the way you're kind of reweighting your evidence is a global way, right? There's not something to examine on SNP is that basically you have to assign the effect of the SNP to one of the SNPs in that region. And because it seems that then some SNPs that may not get above the threshold in a global analysis, if you somehow enforce that, you have to assign the effect to one of the SNPs in a smaller region, that you can discover more stuff there. Does that make sense?
Yeah. Yeah, you're saying that if you had some other evidence that you wanted to be looking at a particular region, you could then.
Well, for instance, you find this global negative association between chromatin and reverse chromatin, right? And the SNP being functional. But if all the SNPs that are in Pickrell equilibrium with a particular SNP, right, are all in repressed chromatin, right? Maybe that SNP is one that activates something.
Oh yeah.
Right? And
yeah.
so that relative scale, it would not be a negative anymore. It's all depending, relative to the other SNPs, can the SNPs in that region, right? What is called original depletion according to your context model.
Yeah. Yeah.
And
No.
then the thing that's locally the most favorable, even if it's globally kind of unfavorable, if it's locally the more favorable context, you would still tend to get a high posterior probability. But it
Yeah.
doesn't seem
Yeah.
in your current scheme.
That's right. The way I think about that is maybe there'd be some interaction term in the model where it's this SNP falls in repressed chromatin, so I should downweight it, but it also falls in some other context, so I should upweight it, and it's some sort of interaction. But I haven't done anything like that yet.
You've done everything independently here, so you've done 18 studies independently. Have you thought about how you could sort of do a meta-analysis, so you capture
Look at
the
all.
sort of GWAS context that's specific to the tissue type or specific to the study, and
Yeah, so what I'm working on right now is trying to look at pairs of GWAS. And so then you can imagine looking at n GWAS at the same time. Yeah, that's